Hi everyone, we will be talking about malaria and the immune system. Malaria overview. Malaria is a disease that is caused by a single-celled parasite belonging to the genus Plasmodium. There are 5 different species of this parasite that can infect vertebrates, with Plasmodium falciparum is the deadliest of the 5. However, not all the mosquitoes have the ability to spread malaria. The transmission vector for malaria is a female Anopheles mosquito. The reason for this is that only female mosquitoes require blood to develop their eggs. Blood contains various necessary proteins and nutrients, such as hemoglobin, which is important for the development of eggs.
The global impact and importance. Malaria occurs mostly in tropical and subtropical areas due to mosquitoes thriving in warm, humid areas. This type of condition allows mosquitoes to develop specifically in ponds and lakes where they lay their eggs. It is considered to be one of the leading causes of mortality in low-income countries. Malaria's significance extends far beyond its immediate health implications, shaping economies, societies, and global health efforts. First, global burden. Malaria affects millions worldwide with an estimated 229 million cases and 409,000 deaths in 2019 alone. Malaria is endemic in 87 countries, primarily in sub-Saharan Africa, Southeast Asia, and South America. Second, vulnerable populations. Children under 5 and pregnant women are particularly vulnerable. Children under 5 account for over 80% of malaria-related deaths. Lastly, economic impact. Malaria imposes a heavy economic burden, costing endemic countries billions of dollars annually in healthcare expenses, lost productivity, and decreased tourism. In addition, malaria places strain on already fragile healthcare systems in endemic countries, diverting resources from other essential health services. Innate immunity, the body's first line of defense against malaria and other pathogens. This type of immunity includes physical barriers, like your skin and mucous membranes, as well as phagocytes or specialized cells, like macrophages and neutrophils, that can quickly detect and destroy pathogens. Inflammation is used in response to infection, using interleukins for communication. Lastly, natural killer cells destroy the infected cells. Toll-like receptors 2 and 9 seem likely the main receptors involved in malaria and central mediators of pro-inflammatory responses during infection. Toll-like receptors display impaired synthesis of pro-inflammatory cytokines when infected with different strands of malaria, impairing the immune system. This graph displays an increased expression of toll-like receptor 2, additionally significantly augmented in CD11. CDs are found on the surface of cells specific to the innate immune system and are involved in adhesion and cell signaling. Interleukins are proteins the immune system uses to communicate and coordinate actions. Interleukins act as messengers between different types of immune cells, telling them when and how to respond to infections. Interleukins can be used in medicine like a conductor of an orchestra to help the immune system play its tune correctly. In this graph, it shows malaria's natural killer cells response were not further reduced by increasing doses of antibodies. It was, however, consistently reduced by interleukin 12 at a concentration of 0.5 milliliter by 50 to 100%.
Now we will talk about adaptive immunity in terms of T cells and how malaria can downregulate T cell responses that makes it hard for individuals to build a robust immune response. To discuss how malaria affects T cells, it is important to first understand the programmed cell death protein PD-1. This protein is located on the surface of T cells and is responsible for suppressing the immune system. When this protein is activated, it triggers a chain reaction that ultimately leads to a decrease in the effectiveness of your adaptive response. Expression of the PD-1 protein on CD4 positive T cells. The PD-1 protein, also known as programmed cell death, is increased in children who live in areas where seasonal malaria is common. In Graph A in this study, researchers wanted to see the relationship between P. falciparum and the potential of circulating CD4 positive T cells. The study was done in Mali, where malaria gets intense. So, in Graph A, it shows PD-1 levels on T cells before and after exposure to malaria. The increased expression of the protein reduces the effectiveness of T cells. To investigate this further, researchers studied the production of cytokines, specifically interferon gamma, TNF, and interleukin-2. They infected 3 mice with acute, chronic, and Plasmodium viruses and then stimulated the cells with ionomycin, a compound that helps scientists observe the production of cytokines and other immune cells. The mice infected with Plasmodium had lower levels of all 3 cytokines compared to the mice infected with other viruses. Cytokines play an important role in regulating the immune response, and in the case of malaria, the downregulation of cytokines make it challenging for individuals to control the disease. So now we're going to talk about T cell proliferation. This refers to the process by which T cells undergo rapid division and expansion in response to encountering an antigen. This process is a critical aspect of the adaptive immune response, allowing the immune system to mount an effective defense against pathogens. In this study, uninfected red blood cells and Plasmodium-infected red blood cells are isolated in a dish to show cell multiplication. In simple, scientists use the assay technique where they mix T cells with the infected red blood cells and added thymidine, which is a radioactive molecule. As the T cells multiply, they grab onto this radioactive thymidine and use it to build new DNA. After a while, scientists check up how much radioactivity the T cells have picked up. More radioactivity means more cell multiplication, showing that the T cells are responding. As expected, the Plasmodium-infected red blood cells were not able to proliferate as effectively as the uninfected cells. The lack of T cell proliferation in individuals with malaria results in impaired immune response that leads to difficulty controlling the infection and increased severity of symptoms and prolonged illnesses.
To summarize, Toll-like receptors display an impaired pro-inflammatory synthesis, suppressing the immune system. Interleukin-12 plays an important role in antigen-specific immunity, or more specifically, natural killer cell response. Malaria parasites can suppress the immune system, which leads to a cascade of regulatory responses from T cells. Upregulation of PD-1 protein on T cells leads to the suppression of vital cytokines, such as interferon gamma, tumor necrosis factor, and interleukin-2. Decreased proliferation compromises the initial response to pathogens and innate immunity, and impairs the ability to mount a robust antigen-specific response in adaptive immunity.